Okay, I think we can start now. Good afternoon, everybody. Welcome to today's EU-OPENSCREEN webinar, Introduction to Chemosensitive Profiling, which is part of EU-OPENSCREEN training activities. So a few words about the EU-OPENSCREEN. EU-OPENSCREEN is a multinational European infrastructure of open screening platforms and chemistry sites for chemical biology and early drug discovery research. You can find more details on EU-OPENSCREEN and its operation through our website, which will be mentioned at the end of the webinar. Thank you all for taking time out and being here today. I'm Kathy Skopelitou, the training manager here at the EU-OPENSCREEN. And in today's webinar, we'll be looking at the following topics. During the first part of the talk, our speaker, Dr. Sheraz will give us an overview of cell-based assay systems. Cell-based assays play a pivotal role in understanding cell physiology and have been an important pillar of drug discovery. Frequently, cell-based assays are used in high-throughput screening of compound libraries. The next topic will focus on how cell-based assay systems can be specifically adapted to chemosensitive assays. Case studies of chemosensitivity assays will be presented during the last part of the talk. I would remind you that this webinar is being recorded and therefore all attendees are muted and the camera pictures are hidden. However, you will have the chance to submit any upcoming questions through the questions and answers icon window throughout the webinar. Of course, there is also the possibility to ask questions anonymously. You can find this Q&A icon on the bottom of your screen in the Zoom window, so if you have any questions, just pop them in there. At the end of the webinar, we will go through the questions, the collected questions, and the speaker will answer them. And if you miss anything, don't worry. We'll be sending around a link to the recorded video when it will be available. At this point, I will ask our speaker to start with his talk, but before we do this, a few words about Sheraz. Dr. Sheraz is the head of the Assay Development and Screening Unit in Fraunhofer Institute for Molecular Biology and Applied Ecology. He has gained extensive expertise in assay development and screening whilst working in academia, the pharmaceutical industry, and currently at Fraunhofer Institute. He has coordinated work packages in assay development, screening, and heat-to-lead studies for many projects. He has co-authored more than 60 peer-reviewed papers and organized 46 global assay development, screening, and drug discovery workshops and trained more than 800 scientists. Sheraz, you may now share your screen with us. We are really looking forward to your talk and sharing with us your expertise in today's topic.
Thanks, Kathy, for the introduction. I will just now share my screen. It's a pleasure to be involved in the EU-OPENSCREEN, Eric, and this is my second webinar. I gave one a few months back with some collaborators from the EU-OPENSCREEN project as well. And today's presentation will cover an introduction to chemosensitivity profiling and these type of assays that are used for various reasons. And as Kathy has already mentioned, there are three main topics that I will go through. First of all, give you an overview of cell-based assay systems, what is typically used in drug screening, also from the clinical side, and for basic research as well. And how can these cell-based assay systems be adapted for chemosensitivity assays? And what is the definition of a chemosensitivity assay? And then go through some case studies as well. And then there'll be a Q&A at the end. So the first 15 minutes will be overviewing cell-based assay systems that are routinely used. But before I do that, I've got a couple of slides on the Fraunhofer Institute. So the Fraunhofer Institute is Europe's largest applied research organization. There are just over 70 sites within Germany, but we also have a number of sites outside as well. And you can see the map in the middle with all the various sites. We are based in Hamburg in the north of Germany, but we also have partner sites across different regions as well. There are roughly 27,000 staff that work within the Fraunhofer, and the annual budget is roughly 2.6 billion euros, a large amount of which comes from industry contracts. I'm part of the Fraunhofer Molecular Biology and Applied Ecology Division, the IME, and we have 600 employees across these sites, which are shown on the right-hand side. A number of these are national within Germany, and we are relatively small in Hamburg, but we have larger sites in Aachen and Schmallenberg, which employ many, many hundred people. And there are 2 international sites as well. Most of what the Fraunhofer does is not life science. We are heavily involved in lasers and optics and informatics and microchips, green energy. But the 600 employees that are shown on this slide are involved in the preclinical drug discovery, and the TMP in Frankfurt. This is actually aligned with a hospital, and they do clinical studies as well. And in Hamburg, our main assets are our high throughput screening infrastructure, which is shown on the bottom row. We have roughly 500,000 compounds stored in assay ready plates for screening. And then we have informatics systems that sit behind all of this to analyze the data, do visualizations, and help us find compounds for progression in the drug discovery value chain. In terms of the drug discovery cascade, you've probably seen this type of slide, more of a cascade from left to right. And bioassays in various shapes and forms are really used in the entire drug discovery cascade, also in chemical biology. And I'll just show briefly where these assay systems come into play. So if you look at, say, a typical drug discovery project on the far left-hand side, before a drug target is found or implicated in a disease for which a bioassay needs to be developed to measure its activity, there's probably 5 or 10 years worth of work that has been done outside industry, in academia, doing basic research, identifying interesting targets which are implicated in diseases, understanding how they work, cloning those targets, developing biochemical assays, cell-based assays. And then once you have an interesting target which you can interrogate or can expose it to, say, small molecules, DNA compound, DNA encoded libraries, or in other type of assay systems, you can really accelerate and begin a drug discovery project. So bioassays take a large sphere of activities pre-drug discovery. Once you've identified an interesting drug target, how do you try and interrogate this activity? You may well purify the protein and develop a biochemical assay with purified protein, or you might develop a cell-based assays. For some drug targets, for example, the GPCRs, which are intracellular and also membrane-bound, in some cases, you may well want to have a cell-based assay using fully functional cells. But for GPCRs, you can also use membrane fragments where you clone and express the target in a cellular environment and then lyse the cells to make a membrane preparation. So here, this is an in vitro biochemical assay, but it does have some cellular component. Having developed the assay and then done some screening, found some interesting compounds worthy of progression, they will then enter various ADME tox assays, cytochrome P450, various cytotoxicity assays, hERG assays to check what is the overall profile of the compound, selectivity assays, and so on. And all of these need bioassays. And as you move from left to right in the drug discovery cascade, cell-based assays become more and more prominent. And then obviously when you are going to get the compound into the clinic, a whole battery of in vivo experiments need to be done as well. And many projects fail to pass various milestones, for example, the hit to lead, the lead declaration, clinical candidate declaration, and even in clinical studies, many substances fail to show efficacy or have adverse toxicity, and they need to be halted. There are also bypass strategies, and we're clearly seeing this in place with COVID-19, where drugs from other compound libraries, other indications are being reused for COVID-19. And remdesivir is one of the front-running compounds that has been approved by the FDA for emergency use in COVID-19, was in fact developed by the pharmaceutical company Gilead for another indication, Ebola. That molecule was literally parked and has now been reevaluated and shown to be somewhat successful for COVID-19. So what types of terms are typically used in the drug discovery cascade? Target validation is very interesting, being sure that the target that we want to interrogate is associated with the disease. And this sounds like a relatively easy thing to do, However, this is probably 5 or 10 years worth of work to establish whether the target is valid. Is there a clear link between the target activity or its malfunction and the disease? And the difficulty is with all of the assay systems that we use, some of which I will highlight, is that these are all reductionist, often using recombinant cell lines, cancer cell lines to overexpress our targets, and they don't really mimic the physiological conditions adequately enough. And it's only when you get your compound into phase 3 in the disease population that you really find out whether the compound is actually efficacious. So for target validation studies, you clearly need a whole battery of assays in place. Compound screening is good to have a complement of biochemical assays where you can find out whether the compound is, say, binding your target of interest, as well as cell-based assays, which would be useful for showing that the compound can enter cells and shows cellular activity. So for secondary assays, we need to have in place a number of assays to measure the in vitro, ex vivo selectivity, liability, cytotoxicity, and all these other activities. And then the compound will then go into animal models, in vivo studies, and then into the clinic. To get a compound to a reasonable point for progression, at least from a drug discovery angle, we have this lead compound criteria, and there are rough parameters that the compound should have properties for. For example, it should be relatively potent, typically sub-micromolar. It should have some cellular activity, and it should have some other properties, which includes should not be too toxic in terms of cytotoxicity, but also, for example, the cytochrome P450s, the hERG liability, and the selectivity as well. The compound should also be soluble and stable in blood. There are no hard figures. We only have some ballpark figures. Typically, if you've got a 1 micromolar compound activity against this primary target, if you have a 10-fold window as a minimum, that is a reasonable starting point for progression. If the window is smaller, then those compounds need to be optimized. You'll find for many of these assays, cell-based assays are being used. For example, the cytochrome P450. You can buy cellular fragments, but you can also measure cytochrome P450 in a whole cell. Cytotoxicity would be used against a whole panel of cell lines, which might be primary cell lines from donors, potentially post-mortem tissue, and also cancer cell lines, which are routinely used. Drugs originating from high-throughput screening have definitely been the case. However, the time difference between FDA approval, in this instance, that's shown on this slide, and when a screen was run, it's typically a decade. Bearing in mind, screening is a very early stage activity where a lot of compound optimization needs to take place. It's typically 10 years before a compound is approved relative to when the screen was run. And oncology is one of the biggest areas of focus for the entire pharma industry. And there are a number of reasons for this. For example, in a clinical trial, you often only need to show progression-free survival. The number of patients that you need in a clinical trial can be relatively small in number. And the price point for drugs for oncology is basically whatever you want it to be. It could be from $10,000 per dose to $500,000 per dose. Phenotypic screening has taken off over the last 5-6 years, where we're using phenotypic approaches, which will be making use of cell-based assays and measuring a phenotypic response. One reason why this wasn't done a long time ago, say 15, 20 years ago, was we didn't really have the technologies to interrogate cellular systems adequately. Now we have high throughput, high content screening systems, and also the image analysis software, which can rapidly analyze multi-parameters, cell morphologies, intracellular events, and be categorized upon exposure to compound. Many drugs have been identified using these phenotypic approaches, where you're just looking for a phenotypic response. This could be as simple as getting a primary cell line, exposing it to compound, and checking if the compound reduces cell proliferation. This may be a starting point for a cancer drug, as long as the compound is not too toxic. And there are a number of drugs which have been shown on this slide. On the bottom right, which were found from phenotypic approaches, where the mechanism of action of these compounds was not known, but they did cause a lack of proliferation, and then subsequent research showed or led to the identification of their molecular targets. However, most assay systems that we use in screening are mechanistic, where we have some understanding of what is going on within the cellular environment. So how do we try and adapt these assay systems for screening and for chemosensitivity assays? So what is the definition of a chemosensitive assay? From the National Cancer Institute, there's a specific definition. It's a laboratory test that measures the number of tumor cells that are killed by a cancer drug. The test is done after the tumor cells are removed from the body, and a chemosensitivity assay may help in choosing the best drug or drugs for the cancer being treated. And the backdrop to this is pretty complex. And historically, when we've gone from identifying a drug target, developing an assay system, which might be biochemical, and then moving on to a cell-based assay and finding a compound which gives a response which is results in the alleviation of a disease. One of the things that we often are unaware of, that these compounds often have unknown activities. They are often polypharmacology effects going on, which we haven't really taken into account. And on the left-hand side, we see staurosporin. This is a relatively potent compound, which is a known kinase inhibitor. And there are roughly 500 kinases within the human genome. And the spider picture that you see in the background is a dendrogram of the various types of kinase classes. We have receptor tyrosine kinases, the RTKs. We have soluble kinases as well, which are shown more in the bottom side. We have those kinases which are part of complexes, for example, the cyclin-dependent kinases. And at the end of each branch is one particular kinase of interest. Staurosporin is a relatively potent nanomolar IC50 inhibitor of many kinases. The middle compound, SB203580, was designed by SmithKline-Beecham and was meant to be a p38 inhibitor of reasonable potency and selectivity as well. However, this compound was designed to be a p38 inhibitor and also for having anti-asthma activity. However, in the clinical trial, there was insufficient efficacy, even though the early target validation studies showed that this compound should be efficacious for asthma. And on the far right-hand side, we see Glaxo-Wellcome's 2016. This is also known as lapatinib, which is a very potent and selective kinase inhibitor for RTK, which is involved in breast cancer. This is an approved drug for breast cancer, lapatinib, and is a very potent and selective inhibitor. However, this Glaxo drug from this picture only shows its selectivity profile for the kinases. There may well be some other inhibition profile of this compound against other target classes, for example, the GPCRs. And we know within the human genome there are roughly 20,000 drug targets. It's very rarely the case that any drug has been comprehensively profiled against every drug target that's available. For some drug targets, we do not have assays. So you could argue, well, that's one reason why we do not have any data, but I am unaware of any compound which has been comprehensively profiled against every possible bioassay for every drug target that we know of. So there are some unknowns around every drug that's been approved. And many of these things that we find out from phase 4 studies, these adverse events where we try and understand what could be the cause of the adverse event. So even though we try and focus drugs to be potent and selective, there are lots of unknowns, and we have to be careful that many drugs do have some polypharmacology effects as well. And this paper nicely summarizes an observation that was found in the clinical trial, and then the retrospective study that was done to understand what is the cause of the polypharmacology and how can we try and alleviate this. So this HDAC inhibitor, panobinostat, was designed to be a potent and selective inhibitor of the histone deacetylase class of proteins. Having a selective inhibitor for the HDACs is pretty tricky because these enzymes have a deep pocket with a zinc inside the active site. So many HDAC inhibitors are zinc chelators, hydroxamic acids, which can go deep into the pocket of the enzyme, and then there's a linker, and then some other moiety of the molecule which might introduce some selectivity. What was found from panobinostat, although it was a relatively potent inhibitor of HDAC, showed some efficacy in clinical trials. There were some adverse events that were reported, and further studies were done to try and understand what could be the origin of the polypharmacology. So panobinostat was derivatized onto saffurose, and pull-down studies were done using various cell lysates. And the research showed that other proteins, for example, PAH, which is phenylalanine hydroxylase, and other protein, the TTC38, the FAD-S2 and FAD-S1, were also binding to the saffurose panobinostat insoluble matrix. It was actually found with further research that the cause of the off-target effect, the adverse event, was actually due to the unexpected inhibition of panobinostat upon phenylalanine hydroxylase. This kicked off another branch of research where a second generation of panobinostat molecules are being designed to try and overcome these inhibition profiles of the non-HDAC enzymes. However, it's also been found that phenylalanine hydroxylase is also implicated in another disease. This led to another project being kicked off where panobinostat was being modified to try and reduce its activity on HDACs, but have an improved profile, an inhibition profile for phenylalanine hydroxylase. So this is an example of a project where a compound was designed for one particular disease, for one particular target class. Clinical studies showed a number of off-target effects, and a lot of research was done to try and identify what was the cause of this. This type of scenario you could really apply to every drug that's on the market. It's derivatization and pull down studies to try and find out what could be binding to the molecule. You could argue, why couldn't this be just done as a standard for most drug discovery projects? I think for those people who are involved in pull down studies, one of the issues is the cell lysates are pretty dirty and they might not be enriched with certain proteins. And the pull down studies often yield very confusing data sets where a lot of proteins are binding to the matrix, and considerable amount of research needs to be done to find out which of those proteins that is bound is actually of interest. Is it just some general precipitation? So having some understanding from the clinical aspect does help. Cell-based assays, what can we do for cell-based assays? A whole range of activities are possible. Even though cell-based assays are not in principle like biochemical assays where we're measuring activity of a particular protein and its substrate, with adequately set up cell-based assays, we are able to look at specific effects and not look at other observations. So this is an example of a cell-based assay where cells are taken from, say, hex cell line, transfected with our kinase of interest and a particular substrate, the IKK alpha, and then we measure its phosphorylation after, say, 1 day, and then you can run this assay system plus and minus compound. There are lots of other proteins within the cellular environment. The kinase might be part of a complex, which is good in this particular case. And using the TR-FRET readout, we are only looking at the phosphorylation of the IKK alpha. Every other phosphorylation event that's occurring within the cell, we're not really measuring. And if you expose this system to compounds, if there's a reduction in the phosphorylation, we can try and correlate that with that compound is inhibiting the upstream kinase. However, The compound could also be cytotoxic, could be inhibiting other kinases within the pathway. So further research needs to be done to find out what is the real mode of action of the compound. But cell-based assays can be set up so that look at specific cellular events without other observations being taking place. For cell-based assays, the assay window typically shrinks. If you can get a 2- or 3-fold window, which has got low CVs, that should give you a good Z prime, which should be amenable to screening and compound profiling. On the other extreme, we can also use the same type of assay on a high content screening platform and do imaging of any subcellular or intracellular event. That could be a particular pathway, an endpoint protein, and you can do multiplexing as well. So one of the earlier papers that was done in looking for small molecule cellular reprogrammers was where cells were plated out and then exposed to compound for a number of days, in this case up to 7 days, and the research group were looking for compounds that could activate the expression of cell differentiation proteins, for example, NANOG, OCT4, SOX, and LIN. These are proteins which are transcription factors which can enable cellular reprogramming. However, with high content screening, the data and the pictures that we get are very complex, and you need adequate software to do image analysis. And there is the Broad Institute in the US, in Boston, where they have a cell painting algorithm, an ImageJ, some of which is freely available and you can download and use for image analysis. And that type of software can be used for any type of high content picture in a JPEG format. But with cell-based assays, one of the issues is the compound activities are often complicated. For example, in this case, we're looking for activators of our transcription factors. They probably are promiscuous. Those compounds which are inhibitors may well be cytotoxic at the same time. So this is another complicating factor of cell-based assays. How do we try and deconvolute the activity of the compound? With cell-based assays, you can do multiplexing, which is a good thing to be doing if you are trying to adapt any assay system for chemosensitivity assays, is to run in parallel a endpoint assay. On the top, we show a time course of AlphaScreen, where we're looking at specific phosphorylation of our protein of interest within the cellular environment. And at the bottom, we show a high content imaging readout at the same time points. And you can clearly see that the two can match together. So this is a good type of activity to do, where you parallel track a biochemical assay or a cell-based assay looking at a specific cellular event, and also high content imaging to allow you to understand what the compound is doing with reasonable confidence. But you also need a good reference compound, a inhibitor, which should be ideally selective, non-toxic, for which a lot of information is available that you can use to probe the cellular system. So biochemical assays are very powerful, and so are cell-based assays in terms of interrogating and understanding how compound mediate the activity of drug targets. It is now relatively standard to run assays in 384 well format in volumes of 10, 20 microliters. 96-well assay transition to 384 is worthwhile and relatively easy to do, but there are very few organizations that actually do 1536 well screening, because the hardware that you need is pretty expensive, and you're also committing a lot of reagent, which costs time and money to make. And the typical cost per well could be, say, €1 per well. So in a 384 well plate, you're committing €400 per well. For 1536, you're committing over €1,500 per plate that's being screened. If you've got 10 plates, that's €20,000 being committed. For biochemical assays, you can do incisive mechanistic studies, for example, enzyme titrations. You can understand how the enzyme functions in terms of substrate turnover. However, with cell-based assays, the substrate turnover and enzyme titration is not really possible. Cell-based assays using endogenous cell lines, primary cell lines, where the targets are being expressed at endogenous levels. You really don't know the actual concentration of protein within the cell. And one of the issues with cell-based assays that use recombinant cell lines, or where you transfect, you are often overexpressing the target of interest just to get a good window, but that might not mimic what's going on in a physiological environment, which you might cause issues later on in your when you try and transition the compounds to in vivo studies. As most compounds are stored in DMSO, DMSO tolerance studies need to be done, day-to-day variability and plate-to-plate variability as well. Once you've developed your bioassay, screen it against a set of compounds, which might be for repurposing studies, a few thousand through to many millions of compounds in the case of big pharma industry. Once you've identified your initial list of hit compounds, they will then be progressed via optimization studies to improve its potency, decrease its liability, toxicity, and then hopefully lead to a lead compound being declared. And I already showed you what the criteria for that would be. In terms of high throughput screening automation setup, there are a number of things that can be done relatively easily. For example, the 96 to 384 well transition is something that should be done. The first row on the table at the bottom shows that the typical assay volume is 50 microliters for a 96 well plate. Miniaturizing this to 384 well screening using fairly cheap automation is definitely worth doing, as that will result in reagent consumption from, say, 6 liters to 1.1 liters if you're doing 100,000 compound screen. However, miniaturizing to 1536, the assay volume per well really doesn't decrease too much, but there's a lot of overhead that needs to go in to make sure that the compounds can be screened in that type of format. As we work in a collaborative environment these days, many assays need to be transferred from one partner to another. 1536 is not commonly used. 384 well assays are probably more used by most partners, but there will be some organizations that use 96 well plates only. So if you've got a 384 well assay, to try and unengineer this into 96 well plates, it's relatively straightforward as well. And what type of assay readouts can we get? I already showed examples of direct readouts from cellular systems using, say, AlphaScreen or TR-FRET. You can also do high content imaging and get very complex pictures. But the way things are going at the moment are to do high content imaging on the fly. So I've got a movie here where we're trying to grow cardiomyocytes in 2 dimensions in 384 well plates. And this is beating tissue, which is technically now easy to do. For roughly 1,000 euros, you can buy a vial of cardiomyocytes. They can be cultured and they will beat in your cell culture flask. However, image analysis around this is still lagging. And on the next slide I show here, this is a hanging drop of a group of cells which are in one droplet of cell culture media on a cover slip slide upside down. And we have imaged this organoid and it does beat erratically. And what is a take-home message from these type of studies? To try and get cellular systems, not just using cell lines, but to try to make them more physiologically relevant. We need to do co-culturing. We need to try and make organoids, these type of cellular systems. However, technically they are easy to do now. However, imaging and analyzing beating tissue on the fly is not easy. This is only really applicable for cardiomyocytes, but if you're doing neurology research where you might culture neurons, as those neurons expand their neurites, you might want to image them, measure the rate of growth of the neurites, see what effect that compounds have on the growth rates. You need to have adequate image analysis software. And Sony have developed this high content imaging platform, which has come out of the TV industry. And effectively what they do here is to culture cardiomyocytes in this case, and as the cells beat, the imaging software will draw a grid above the beating tissue, and as the tissue or cells expand and contract, the grid that's superimposed on the cells will also contract and relax at the same time. And with the 200 frames per second camera within this device, you can almost get an ECG type of readout, which is shown in the middle. So this beating cardiomyocyte tissue, as the cells contract and relax, the software can analyze on the fly how what effect compounds are having on the beating of the tissue. This is important for understanding potential cardiotoxic effects of compound in a cell before the compound goes into the clinic. So some of the case studies around chemosensitivity assays. I've covered briefly the type of assays that are used in drug screening, and they are very similar to what's used in chemosensitivity assays. And when you are doing your project, say in the case of drug discovery, one of the key things that you will do is to show in a preclinical animal model that you can see shrinkage of tumor without too much gross toxicity. And this is almost like a mini proof of concept study in an animal model, a xenograft model, where you might have a rodent, where you have implanted a tumor, and then you expose the rodent to the compound. And on the top left-hand side, you see the shrinkage of the tumor in the presence of the drug, whereas in the case of the vehicle, no drug, there's an increase in the tumor size. And on the bottom left, the tumor size, although it's shrinking in the presence of the drug, the overall mass of the animal stays relatively stable, indicating that there's no gross cytotoxicity being observed. This is the readout for a project on cancer for where compounds that were inhibitors of these EZH2 drug target were synthesized. However, this compound has been claimed to be ready for clinical studies, at least phase 1, to check its toxicity in humans. But to get to this point where you can show an in vivo proof of concept, it's typically 5 years before you can really get a compound that is clean and safe and not too toxic and doesn't have all the other side effect profiles as well. And how have chemosensitivity assays progressed? Effectively, chemosensitivity assays are where you take biopsies, cellular material from often ill patients, typically cancer patients, and then you measure the activity of cellular events, either using the techniques that we already have, and I've already shown, for example, AlphaScreen, which can be used to measure activity of many, many drug targets, or there may well be TR threat readouts, but essentially what you are doing is you are getting tumor material from the patient, from the human individual, rather than making the recombinant cell line. And chemosensitivity assays have moved on considerably from the 1970s up until relatively recently, but they all involve obtaining tissue samples from patients and measuring various effects. The AlphaScreen technology has been around relatively recently, maybe for 10, 15 years. Prior to then, it would have been very difficult to measure, say, intracellular phosphorylation events easily. The MTT assay to measure cytotoxicity is still used extensively, especially in academia, which is robust, reliable, and accepted by all researchers involved in cytotoxicity assays. But one of the key things around the MTT assay is that it was used extensively to check what compound should we be giving the patient for, say, as an anti-cancer treatment. So you could give the drug as part of a clinical study at risk to the patient, as you probably don't know what adverse effect it might cause on the patient, obtaining some tissue from the patient and exposing it in a cell-based assay to measure the effect of potential compounds or therapeutics, either in isolation or as combination, on the patient itself, outside the patient in a test tube, would allow you to gain some understanding whether that compound will be efficacious or whether it should be given to the patient, because you've got to balance the risk-benefit profile. These studies are still being done where tumors are taken, biopsies are taken, pathology is typically done to understand what protein might be overexpressed within a particular tumor. For example, we know in the case of breast cancer, there are certain genes which are upregulated, and if those genes are present, you can be sure that this drug should be given. In the case of patients where certain genes are not upregulated, for example, the BRCA1 and A2, then you might not want to give them that specific treatment. And if you look at the breast cancer survival rates over 10 years, we have gone a long way in improving the survival rates over 10 years. And however, there's still a long way to go. On the left-hand side, 50 years ago, the combination of the CMF, which is still used today as one of the initial therapies for breast cancer, is still being used, which are very nasty drugs, lots of side effects, hair loss, significant loss in mass and appetite, and all the other side effects. And we have found other molecules and also antibody drugs which have increased 10-year survival rates considerably. However, we've still got a long way to go. And how can we try and improve survival rates further? The chemosensitivity assays will play a key part in this, and also other biomarker and diagnostic tools. If you look at the breast cancer and BRCA1 and A2, these are tumor suppressor proteins. We now know, and in the US, there was a time when before a certain drug was being given, which was relatively expensive, this diagnostic test would need to be done to ensure that the patient either has or doesn't have specific mutations. That would allow the doctors to triage the patients and ensure that the patient would be given a drug that would work, preventing potential side effects from giving them a drug that would really be of no benefit. Drug resistance is also a large thing that's coming into play. Many people who have a breast cancer, prostate cancer, pancreatic cancer, once they are given the drug, there's a good response in terms of tumor shrinkage, and then the tumor, it will either mutate or it might undergo metastases, appear in a different part of the body. And we now know tumors are also very heterogeneous. Obtaining this tumor material at regular intervals, exposing this tumor material to different combinations in vitro outside the patient to try and get some understanding which combination of drugs, what dose of drugs would be of benefit to the patient, which is effectively what the chemosensitivity assays involve. Although we've seen the most successes with breast cancer, many other cancers are way behind in terms of overall survival rates. If you look at pancreatic cancer, the disease-free fraction and the survival rates is very low, typically 3-4 months. And then upon exposure to compound, you may survive for, say, up to 400 days. And many studies have been done to try and allow the identification of what drugs should be given to patients with pancreatic cancer. These may well include combinations or also drugs in isolation. And on the right-hand side, this is an average of IC50s of the various drugs, either in combination or in isolation, in ex vivo chemosensitivity assays. So tumor tissues were taken from different cancer patients and then profiled against the compounds. Clearly for those compounds which the IC50s are very high, that compound might not be worth giving to that particular patient. So patient stratification, personalized medicine, this is also part of the chemosensitivity aim. Drug resistance is a big issue. In most cases, drugs are given, there's an immediate shrinkage of the tumor, and then there's a relapse. And adaptive clinical trials, blended clinical trials, these are now being rolled out especially in cancer studies, where rather than having initiating a clinical trial with a fixed protocol, with a fixed readout endpoint. You can do a blended clinical trial where, as the clinical trial is being run, if there's a clear observation where some people are benefiting from a particular drug or a particular combination, you can adapt the clinical trial on the fly. But clearly there are issues in terms of ethics and guidelines. This all needs to be carefully considered when doing these type of studies. Precision cancer medicine, next generation diagnostics, this is the way we are going, also including artificial intelligence, computational tools, where we can take small amounts of tissue and even sequence the genomes of individual cells within a tumor to find out how we can try and get rid of the cancer at an early on stage with minimal side effects. However, when you look at the guidelines, there are no firm guidelines, from what I understand, what should a chemosensitivity assay look like. Often these are unregulated, not approved tests where medical staff are obtaining biopsies from cancer patients. Then they are given to various researchers for understanding mechanism of action of compounds or combinations. And there's still a gap between whether these assays that are being used for chemosensitivity should really be used to decide on what compound should be given to a patient, because it could be a life or death matter in some cases. CAR T therapy, this is one of the drugs which costs, list price is $500,000 per bag, which you see at the top, where blood cells are taken from the patient themselves. They are re-engineered and then reintroduced into the patient. The success rate for this for leukemia is only 5%. So you wouldn't really want to give this drug to every patient. And part of the patient stratification studies include chemosensitivity assays, where we take the blood cells from the patients and gain an understanding what is the likelihood of them actually benefiting from the CAR T therapy. And this is still an emerging field. So artificial intelligence, we can do single cell sequencing within tumors and biopsies. We can also do preemptive studies, take samples from an otherwise healthy person or someone who has got a family history of a particular disease. There are tools that are being developed where we can try and get some sort of prediction on what is the likelihood of someone getting, say, cancer in the next 10 years. And we know one famous example, Angelina Jolie, who underwent drastic surgery because she had a particular mutation. She didn't have the cancer, but she underwent surgery because she thought that she would end up getting cancer at some point because she had that particular mutation in her cells. So in conclusion, it's an exciting time for drug discovery. Most of what we want to do, we have concepts of, but the tools and technologies haven't been there to allow us to really probe intracellular events, even down to specific cell lines with specific tumors. And the recent papers come out in Science where they have used CRISPR-Cas to actually eradicate entirely in an animal model, although cancer. So they made a xenograft model and then used CRISPR-Cas to actually edit out the gene, which is the cause of the cancer, and they claim to have completely eradicated the cancer for those particular animals. Whether this will translate to humans, we'll have to wait and see. But CRISPR-Cas, we know it won the Nobel Prize for Chemistry this year, and it's a very powerful technique. Whether it will be possible to be used or allowed to be used on humans, that's a separate issue. But it's collaboration is also key to success. There are lots of things that we want to do, but we are all experts in our own little fields of research. And to get a full understanding of chemosensitivity assays and other things, we really need to work in a collaborative form. And we also need to interrogate historical data, going back into patients who might have been given certain treatments, who either survived for longer or died earlier. This is all useful information that we need to try and interrogate as well. And again, this is where all computational tools come into place. So that's where I end. So thank you for listening, and I'll be happy to take some questions.
Thank you very much, Sheraz, for your engaging talk. This was indeed a very interesting and up-to-date overview on chemosensitive profiling and chemosensitivity assays, as well as the trends and challenges in this field. For those of you just joining us now, welcome to today's webinar. I would remind all of you that you can type your questions in the Q&A icon window at the bottom of the Zoom window. So I guess it is now time for your questions. So our first question to you, Sheraz, what is the cost of developing a cell-based assay?
Yeah, so often if you are doing drug screening and you're screening large numbers of compounds, which is what the big pharma industry do, this has a huge cost implication. And what we typically use is measure is the cost per well. For a biochemical assay, we try and aim for a cost per well of 10 cents, 0.1 euro, which might not sound like a lot, but if you're screening 1 million compound, that's a huge amount of money. If you're doing drug repurposing of, say, 10,000 compounds, then you can, say, have an assay cost per well of, say, €0.5 or €1. For cell-based assays, they are more expensive. Starting price is really between €0.5 upwards. And if you're doing multiplexing and buying specific reagents from certain vendors, this can cost 2, 3, maybe €5 per well, which is very expensive. But if you go and buy a kit from a particular vendor, a typical cost of a kit is between €500 and €1,000, which will give you 1 microtiter plate. So running assays and the cost is not cheap.
Yes. And how long does it take to develop a cell-based assay?
Many assays that we use, kits are available from many of the big vendors. But I've already mentioned that the price per kit is roughly €1,000, but time is money as well. If you want to try and develop an assay from scratch, that could take many months because you've got to make the cell line, the protein. If you're doing an antibody detection, you can go and buy the antibodies and then do coupling with floral labels yourself. But this all costs time and will result in a delay in the project. So we find generally as a first start, it's good to just buy a kit at relatively low cost, say €1,000, just to do a proof of concept study. Is the idea working? And then you might want to develop your own reagents. But generally the trend is going towards buying the kits rather than developing assays from scratch.
Another question from one of our participants: how many selectivity data are required to move a compound onto the next phase of optimization? And is it more important to look for similar targets in the same target family, or better to look for activities in unrelated target families?
Really, both of those things that have been mentioned. There are companies out there which do comprehensive compound profiling. If you are doing a project on one particular target, it's very unlikely and it's not really reasonable for you to develop the whole panel of assays. You might develop 1 or 2 assays as part of your project understanding. So if you're doing, say, a kinase from one particular family, you might also develop 2 or 3 others internally. But in most cases, companies typically just go to CROs that offer these as a service. So you can buy access to a kinase panel where you just give them your compound and they will give you a full kinase profile or a full GPCR profile. To develop these assays in-house is really unrealistic and it's not really worth doing. It's just too expensive, too time consuming. The problem with drug discovery in general is we are working in silos. Many compounds will fail in the clinic, and the statistics are pretty stark. More than half of the compounds that go into the clinic fail because of lack of efficacy or high toxicity. So in terms of the toxicity assays, you need to get these done as much as possible. And there are probably 5 or 10 of these assays looking at toxicity in primary cell lines from different parts of the body, hERG, cytochrome P450. But looking at off-target effects, this is really a huge endeavor. This is unrealistic, and really you need to try and tap into your collaborators who might have these other assays where you want to gain an understanding. But to do one target class from another family might not be enough, because there are lots of, if you've got a kinase inhibitor and you test it against one GPCR and it's inactive, it doesn't mean you've ticked the box. Okay, it's not active against that GPCR, but there are 300 that you need to test them against. And this all costs money. I mean, it's possible, but to do a comprehensive compound profiling will cost hundreds of thousands of euros for just for one compound.
A very interesting question coming from one of our participants. You showed 3D models to do chemosensitive assays like hanging drop organoids, etc. What do you think about microfluidic systems allowing for drawing microtissues from patient samples? Could you comment on that technology?
Yeah, I didn't mention it in my presentation, but there's a huge field of research around microfluidics, organ on the chip. All of this is being done where people are trying to make artificial hearts, artificial brains, artificial livers, co-cultures on microfluidics chips to try and miniaturize the amount of material that's used. But remember, the material that you get from the patient is of limited supply. There are only a certain number of assays that you can do. And then once you've got that data set, obtaining another sample might be difficult. And even if you can, the results might start to differ from what you have already observed. One of the issues with microfluidics is it sounds good in principle, as do what I've shown in terms of the 3D organoids and the beating tissue. To really interrogate this assay on the microscope and do 3D plots and really understand what's going on is very, very difficult. In principle, microfluidics is good, but it's very, very challenging to use and implement.
Another question regarding the ethical issues. Do you see formal ethical issues in obtaining samples from patients in clinics for chemosensitivity? Is it realistic to expect it that it will become a common routine in the next 10, 15 years?
I would have thought so. I think patients are also becoming more clued up as to clinical trials, what do they entail, and many patients for whom there are no drugs, the orphan diseases, or patients who have been through various types of chemotherapy and hasn't really ended up working entirely or curing the disease. There are many people who are very astute, are aware of where things are going. And I think the corona has also opened up the Pandora's box where people are really questioning the layperson. The average person on the street is also asking, what is this all about? How can we, what are the next generation therapies? But I think the doctors are also being clued up with the technologies and the tools that we have. So I think it's a collaborative effort. And I think patients who already signed consent forms, most of them probably don't even read what they've signed up for. So it's a tricky issue, but there will be, I think, further expansion and acceptance by patients that they are giving their samples for testing and giving them some personalized treatment.
Yeah, and one last question, since we are running out of time. I think you mentioned this during your presentation, but do computational methods and artificial intelligence fit into chemosensitivity assays?
I think the AI and machine learning is being applied to everything. So it's probably at a very early stage here in terms of drug screening and chemosensitivity, but there is a lot of historical data there. And it's a question of maybe just interrogating the historical data that we already have for patients who've undergone treatment and then seeing how we can move forward with this. So this is still at a very early stage.
Okay, so I think we're running out of time. So thank you again, Sheraz, for your very interesting talk. And thank you all for your questions and answers. At this point, and before thanking you all for being here today, I would take this opportunity to tell you that we would love to hear back from you. So after the end of the webinar, a feedback questionnaire will be sent to you by email. Your feedback and comments are more than welcome in order for us to improve ourselves and advance our future training activities. If you would like to receive more information on EU-OPENSCREEN ERIC technologies, partner laboratories and services, or simply subscribe to our newsletter, please don't hesitate to sign up using the link indicated in the blue arrow, or just scan the QR code. If you want to know more about the EU-OPENSCREEN, you can visit our website, www.euopenscreen.eu, or contact us through our Twitter, Facebook, and Instagram accounts. Thank you very much, Sheraz, once again, and thank you very much all for joining us, and wish you a very, very nice evening. Bye.